The second lecture is devoted to classical genetics. We will describe three questions: the first and the second Mendel's laws, the third Mendel's law, and limitations of Mendel's laws. Before the 20th century, people believed that biological traits passed from one generation to the next through the blood. Many people still speak of blood relatives. However, we now know that biological traits are passed from one generation to the next through chromosomes in the parent's sex cells. Genetics is the study of heredity and variation of organisms. Basic principles of genetics were first discovered in the 19th century by Gregor Mendel, an Austrian monk who performed experiments with pea plants. Mendel's findings were published only in the little-known magazine. His work did become well known in the year 1900 when Correns, Chermak, and De Vries performed breeding experiments independently and obtained results similar to Mendel's findings. Mendelian genetics. Since his family was poor, he entered a monastery to obtain an education. He became a priest at the age of 25 and studied mathematics, botany, and other sciences at the University of Vienna. Later, at a monastery in the town of Bern, Mendel worked on plant breeding experiments. He was interested in learning how plants inherit certain traits. From the results of his experiments, Mendel was able to explain some basic principles of heredity. Today, these principles are referred to as Mendelian genetics. The first question: first and second Mendel's laws. Mendel's scientific approach. Mendel studied heredity in a scientific manner by conducting carefully planned experiments. He decided to work with garden pea plants, which grow quickly and are easy to cultivate. Normally, the flower's petals remain closed until the plant self-pollinates. But Mendel pried open the flowers and removed the male pollen-bearing structures from the flowers to be pollinated. This procedure prevented female egg cells in a plant from becoming fertilized by male sperm in the pollen from the same plant. Then, Mendel selected the plants he wanted as parents by cross-pollinating the plants as shown below. In this way, Mendel controlled the way the plants were fertilized. The first, Mendel worked with only one trait at a time. He studied only those traits that were easy to see or to measure. For example, some of the traits Mendel studied included flower color, seed shape, and stem length. By working with one trait at a time, Mendel could more easily interpret the results of his experiments. The second, Mendel kept detailed records of his observations. He counted large numbers of offspring and looked for mathematical relationships based on how often specific traits appeared. Mendel began his experiments using strains of plants that bred true. This means that through the generations, these strains always produced offspring whose traits were identical to those of the parents. Mendel was curious to know what would happen if he crossed two strains of pea plants that looked different. Which trait would the offspring inherit? In one of his experiments, Mendel crossed pea plants that had red flowers with pea plants that had white flowers. Pea plants that inherit an allele for red flower color from each parent will be a pure strain, or purebred, for red flower color. Offspring will be purebred for white flower color if they inherit an allele for white flower color from each parent. Mendel's results led him to conclude that traits, such as flower color, are determined by individual units, which he called factors. Today, these factors are known as genes. Mendel noted that the results of his experiments were best explained if each offspring receives one factor, gene, for a trait from each parent. These genes are passed to the offspring through the parent's sex cells, or gametes. Since the male and female gametes unite during fertilization, Mendel reasoned that each trait in an offspring is determined by a pair of genes. The two genes that make up the pair, however, can come in different forms. For example, the gene for flower color in pea plants comes in two forms. One form produces white flowers, the other produces red flowers. Today, the different forms of a gene are called alleles. A pea plant which inherits an allele for red color from one parent and an allele for white color from the other parent will be a combined strain or hybrid for flower color. When Mendel crossed purebred red-flowered plants with purebred white-flowered plants, he observed that all the hybrid offspring in the first generation, called the F1 generation, had red flowers. Even though each offspring received an allele for red flower color from one parent and an allele for white flower color from the other parent, only the red flower allele was expressed. Mendel concluded that red flower color was dominant over the white flower color. White flower color was recessive, since it was not expressed in the hybrid offspring in the first generation. The word dominant comes from the word dominus, in Latin, master. The dominant allele is expressed in a hybrid. The recessive allele is not expressed in a hybrid. Scientists use symbols to distinguish dominant and recessive alleles of a gene. Notice in the diagram that the symbol for the dominant allele is written as an uppercase letter. The symbol for the recessive allele usually is the lowercase of the same letter. Thus, the dominant allele for red flower color is written as big R. The recessive allele for white flower color is written as small r. The possible combinations of alleles for flower color in a pea plant are represented as big R, big R, big R, small r, and small r, small r. Homozygote is an organism or cell having identical alleles of a particular gene and so breeding true for that gene. Heterozygote is an organism or cell having dissimilar alleles for a particular gene. For example, big R, small r. This picture represents the scheme of cross from the first Mendel's experiment. Purebred parents are crossed, only one type, one kind of gametes is produced by each parent. Hence, only one hybrid organism is possible that possesses two different alleles, dominant allele big R and recessive allele small r. Mendel's law of dominance can be stated as follows. When an individual is hybrid for a certain trait, only one of the two alleles is expressed. The allele that is expressed is the dominant allele. In some English books, we can find another definition of Mendel's law of dominance. This definition describes an experiment. As a result of crossing dominant and recessive homozygotes, all descendants are formed identical. They are heterozygotes. Mendel did not observe any white flowers in his F1 generation hybrid plants. Could the recessive allele for white flowers have been lost? Mendel discovered the answer to this question by crossing hybrid plants from the F1 generation to produce a second generation of offspring. After examining the offspring in the second generation, or F2 generation, Mendel found that approximately 75% of the plants had red flowers and approximately 25% had white flowers. The recessive allele for white flowers was not lost or changed in the hybrid plants after all, since white flowers reappeared in their offspring. The two alleles for flower color in the hybrid plants, big R, small r, must have separated or segregated when the hybrid plants formed gametes. Each gamete received only one allele, big R or small r, from the pair of alleles. Mendel's law of segregation can be stated as follows. The two alleles that determine a particular trait will segregate when sex cells form. Half the sex cells will receive one allele from each pair of alleles. The other half will receive the other allele of the pair. In some books we can find another definition of this law. This definition describes the experiment. At crossing of heterozygotes, we can observe segregation of signs or segregation of traits in offspring in the ratio 3 to 1. The second question describes the third Mendel's law. Mendel performed breeding experiments for about 12 years, during which time he grew thousands of plants. In all, he studied 7 different traits in pea plants. For example, he studied pea plants in which round seeds, dominant trait, big R gene is responsible for this trait, were dominant over wrinkled seeds, small r, and yellow seeds, big Y, were dominant over green seeds, small y. Mendel wondered whether the inheritance of one kind of trait influences the inheritance of another trait. For example, does the inheritance of seed shape affect the inheritance of seed color? Mendel began studying how different traits affect each other by crossing a purebred strain of plants that had round, yellow seeds, big R, big R, big Y, with a purebred strain of plants that had wrinkled, green seeds, small r, small y. The diagram shows that all the offspring of this cross had round, yellow seeds. Based on his previous results, Mendel suspected that the offspring were hybrid for both traits, big R, big Y, small y. Organism with 2 hybrid traits is called dihybrid or diheterozygote. Mendel continued his experiment by crossing the plants which he suspected were dihybrid for seed shape and seed color, big R, big Y, small y. Mendel crossed 2 dihybrid plants and analyzed the results of his experiment. He found that 9 of 16 plants were round yellow, 3 of 16 were wrinkled yellow, 3 of 16 were round green, and 1 of 16 were wrinkled green. He concluded that alleles for seed shape and color segregated independently of one another. Each parent produces 4 kinds of gametes: big R, big Y, big R, small r, big Y, and small r, small y. The Punnett square shows that the combinations of these gametes would produce 4 different kinds of offspring. After making the crosses, Mendel sorted and counted 556 seeds that were produced by the crosses. He found that 315 seeds were round and yellow, and 101 seeds were wrinkled and yellow. A total of 108 seeds were round and green, and 32 seeds were wrinkled and green. The offspring nearly fell into a 9 to 3 to 3 to 1 ratio for seed shape and color, as Mendel had predicted. Since the experimental results were close to the results he predicted, Mendel concluded that different traits segregate independently of one another when sex cells form. The third Mendel's law is called law of independent assortment. This law can be stated as follows. The inheritance of alleles that control one trait is not affected by the inheritance of alleles that control a different trait. Alleles that control different traits segregate independently of one another. Another definition describes the experiment. At crossing of 2 dihybrids, we can observe segregation of traits with different combinations in offspring in the ratio 9 to 3 to 3 to 1. Mendel formulated the basic principles of heredity without really knowing what was inside the sex cells of the pea plants. He didn't know where the factors or genes were located. Since Mendel's time, scientists have learned that genes are located on chromosomes. The behavior of the chromosomes determines how the genes are inherited. Walter Sutton noticed that the behavior of chromosomes during meiosis was similar to the behavior of the genes Mendel studied. He guessed that the genes Mendel studied were located on the chromosomes. The third question: limitations of Mendel's laws. Pay attention that Mendel's laws apply to eukaryotic organisms that reproduce sexually. Hence, Mendel's laws doesn't work in bacteria. Mendel's laws are statistical in nature. It means that the real ratios of phenotypic classes among the offspring deviate from the expected ones within the statistical error. The chi-square method is used for analysis. The first limitation. The hybrid forms all types of gametes with equal probability. Heterozygote organism big A small a produces 50% gametes with dominant gene big A and 50% gametes with recessive allele small a. The second limitation. The combination of gametes during fertilization occurs with equal probability. Each egg with dominant gene big A or small a, different sperm cell with dominant gene big A or recessive gene small a. The third limitation. The same viability of zygotes of all genotypes BK small a, and small a small a. All organisms will survive. But in some cases we can find the presence of lethals. Lethal alleles or lethals cause cell death or death during embryogenesis or immediately after birth or at any early age before puberty. Semi-lethal alleles also exist. They cause the death of more than 50% of individuals, but not all. Pay attention to the example of such trait that is caused by lethal allele. Achondroplasia is an autosomal dominant disease in which the growth of long bones stops prematurely, but all other bones grow normally, so a person develops dwarfism. Dominant gene big A is responsible for achondroplasia, dominant trait. Homozygosity for this gene is lethal, and such children usually die in utero or at an early age. Recessive allele small a determines normal growth. Suppose we study the cross of two dwarf persons. According to Mendel's law, 25% of children must have the genotype big A, 50% must be heterozygous, big A small a, and 25% must be recessive homozygous persons. But indeed, homozygous persons will die, and in the F1 generation we can find the ratio 2:1 instead of 3:1. Conclusion. If lethal allele is present, Mendel's ratios are modified. This gene for achondroplasia is very interesting. It is dominant gene when determines dwarfism, and this gene is recessive if determines lethal effect, because only homozygous persons will die, persons which have two identical alleles, big A, but such situation describes recessive allele. Hence, this gene has special name, dominant allele with recessive lethal action. Other limitations also are described. The fourth, full manifestation of the sign regardless of environmental influence. The fifth, genes are localized in the nucleus, not in mitochondria or chloroplasts. No mutations, no gene mobility. Mendel's laws do not follow in the case of genomic imprinting. Lack of gene interaction, allelic or non-allelic. The ninth limitation. The third Mendel's law is valid for genes located on non-homologous chromosomes. It does not apply to linked genes located at a short distance from each other. Gene interactions and gene linkage will be described in the next lectures. The tenth. Incomplete penetrance changes Mendel's ratios. Also, this very interesting phenomenon will be described in future lecture.